Hello, everyone. I hope you're having a really interesting meeting. I'm delighted to have been asked by Jill to give a little overview of genomics of FAM177A1 and to explore the gene in a little detail in some of the publicly available browsers that you will then be able to go off and try yourselves. So a little introduction. I've got some slides, so if I just share them with you. So I hope you can see my slides. So I'm Dr. Charles Steward. I'm head of patient and participant engagement at Genomics England. And I'll tell you a little bit about them in a second. But first of all, so I've spent, it would be nearly 30 years now working on the human genome. Most of that was at the Sanger Institute on the Wellcome Genome campus near Cambridge in the United Kingdom. And I spent a lot of my time actually going through the human genome, identifying where genes were or genes are, and assigning a function to them, trying to understand a little bit more about what they do and whereabouts in the body they're expressed. And I published work in a number of papers, but I've listed three sort of key areas that I've been working in, and that's on chromosome 10, which was published back in Nature in 2004. And then did quite a lot of work looking at type 1 diabetes in mouse. And then for personal circumstances, which I'll tell you about in the next slide, I then started to look at genes specifically involved in neurological disorders, such as epilepsy. And more recently, I'm beginning to look at genes involved with cerebral palsy. I did my PhD at the Sanger Institute, and Sanger is most famous for generating around 1/3 of the human genome, which is the largest proportion of the human genome by any institute. And I live in the Black Forest. Currently moved here for personal circumstances, but it's a lovely place. They make cuckoo clocks, they make lovely cake, and it's also where Hansel and Gretel comes from, the fairy story, which I'm sure you all heard of, actually based on some truth from the 14th century when there was a European-wide famine and people used to take their children into the forest and leave them there. Anyway, so as I mentioned, I work at Genomics England, and Genomics England is a company that was set up by UK government to sequence the genomes of around 100,000 genomes of people with rare diseases and cancers. And it was actually set up by our ex-Prime Minister David Cameron here on the left. And the little boy next to him in the wheelchair is his son, Ivan Cameron, who had cerebral palsy and a type of epilepsy called Ohtahara syndrome. So in many ways, the 100,000 genomes, Genomics England, is a result of poor Ivan's struggle with his very severe neurological disorders. And sadly, he died around the age of 6. On the right, that's me, and that's my wife, Svenja, and my two children, Imogen and Jasper. Both my children, our children, have cerebral palsy and epilepsy. And they are the reason that I'm doing the job I am at the moment. Anyway, moving on. So it's a very basic overview. I believe I'm speaking to people who have some experience in genomics, perhaps some who don't have quite as much. So I thought let's start at the very beginning. So the human genome, what is it? Where does it sit? Well, humans are fantastically complicated, and we believe there are around 37 trillion cells in the human, and we don't know what a lot of them do. And there are roughly around 20,000 genes in the human genome. The count is slightly less at the moment, but it fluctuates as our understanding of biology changes, and we can interpret more of the genome. And genes are responsible for making proteins, which are the building blocks of life. And the human genome set out to understand exactly what the genome consists of and where those genes are. And on the right, there's this picture of, you see the black and white, what we call a karyotype. And these are the chromosomes that make up the human genome. And you can see there are numbers there from 1 to 22. And those are the autosomes. And they're all in pairs. So we each have 2 pairs of the chromosomes, chromosome 1 to 22, and we inherit one from mum and we inherit one from dad. And on the right, you can see there's an X and a Y and a little MT. So we inherit, obviously, we inherit 2 from mum and dad. Sometimes we'll inherit an X and an X, so that we're a female, or an X and a Y, so that we're a male. And the little MT on the side, that's the mitochondrial genome. And I'm sure many of you have heard about mitochondria. They are responsible for energy production and are often associated with different types of disorders, some of which are epilepsy. And the genes are interspersed across our chromosomes. And in fact, genes actually only make up roughly 1 to 2% of the genome. The rest is often referred to as dark matter or non-coding. But actually, we're beginning to understand that this so-called, what people call junk DNA, actually seems to have a really important function in controlling when genes are expressed and how genes are expressed. You know, are they express more or are they express less? So this is really big. You know, it's a really fascinating area for people to begin to understand. So, what is the genome? Well, we often think of it as letters, and 4 different letters in many, many different combinations, and around 3 billion of these letters. And these letters are, as I said, there are 4 of them, and we give them the letters A, C, G, and T for the different chemical nucleotides that they represent, as you can see in my slide. And the order that these letters appear in is what we refer to as the DNA sequence. So many of you will know that, heard of the terms panel sequencing, whole exome sequencing, and whole genome sequencing. So panel sequencing is we just look at a specific set of genes. So there might be sort of 20 genes we're interested in our disorder, and that's all we look at. More recently, people beginning to look at whole exome sequencing. And this looks, as I said, to around the 1 to 2% of the genome that's actually involved in making proteins. So this is much more comprehensive than panel sequencing, but of course it still leaves out a huge proportion of the genome. And whole genome sequencing is where we look at the whole genome. We look right the way across it to see if we can find bits that have changed in a person compared to the reference genome, as we do in panel sequencing, whole exome sequence, but we look across the whole genome. So we, as I said, we don't understand what all of the genome does yet, but if you have a whole genome sequence, you can go back and look at it in 5 years, in 10 years, in 20 years' time, as our understanding of the genome increases, and we begin to understand what the other bits of the genome do. So there are various different types of mutations, although we don't tend to use the word mutations now, but you can think of mutations or variants. And they, on my slide, we have an illustration of what we call a single nucleotide variant, and that's when one letter gets changed to another. Indels, which is short for insertion or deletion. So that's when a bit of the patient genome either is deleted with reference when compared to the reference, or a little bit of extra DNA happens to get inserted. And of course, if this happens in a non-functional part of the genome, it doesn't tend to matter so much. But of course, if it's in a gene, a gene sequence, and then that can be, it can be quite severe sometimes. Or and likewise, so those are sort of the small variants, and then we can have really, really large variants, which we see either sort of the whole chromosome can be deleted or copied, or just sort of large sections of the chromosome. So we have here, if you see an image of a deletion where the orange is removed, a duplication where a piece of the genome is doubled up, an inversion where the sequence actually gets turned around compared to in a reference genome, and an insertion, so we get an extra bit of DNA inserted into our chromosomes. Sorry, what I haven't said is these are representations of chromosomes. And translocations, where a bit of one chromosome gets transferred to another chromosome, and likewise. So you can see there are lots of different ways that variants can occur, or different types of variants, and they can, in some cases, they don't seem to have any impact on a person, but in others they can be they can have a very detrimental effect. So if we're thinking about FAM177A1, we think of it as being what we call autosomal recessive, and there are often we think about autosomal dominant disorders and autosomal recessive disorders. And where we're thinking about autosomal recessive, you require both sets of chromosomes to have a variant of impact. So if we look at the picture over here, you see we have mum and dad. So as I said, we have two pairs of each chromosome that we've inherited from our parents. And dad here, he has one healthy copy, and he has one copy of the chromosome that has got the impacting sequence on it. And that's okay because it's autosomal recessive. It means that there is enough protein essentially being made from the healthy copy that it can compensate for the chromosome that has the variant in it. You see mum here, she too has two chromosomes here, one of which has the normal copy and the one that has the affected copy. And when they have children, there is the odds of having a child with both copies of the variant is 1 in 4. So if you can think, you can either inherit this one or that one from dad, or this one and that one from dad. So if you think about the permutations, you can have this one from dad, this one from mum, and then this one from dad and this one from mum, which would result in an affected child. So you can see the affected child has unfortunately inherited one chromosome with a defunct copy, and this one here from mum, which is a defunct copy. And this is how we think about it for FAM177A1. So what do we know about FAM177A1? Well, we're now going to have a little look in the genome browser and see what we can learn. Excuse me while I get my genome browser up. So, what do we know about FAM177A1? Well, first of all, the name. What does the name mean? Well, when we were originally generating the genes in the human genome back in the late 90s and early 2000s, we didn't know what they did. Most of them, we just had no idea what they did. And sometimes we could look across conservation in different organisms, such as in mouse, perhaps where more experiments have been done. Obviously, it's much easier to do experiments in mouse than it is in human for obvious reasons. But sometimes there was just nothing we knew about them. And so we would try to give as much information when we were generating these genes, even in the case where we knew nothing about a gene. And sometimes genes are members of families. So there are groups of genes in the human genome that have similar sequences with others. So if we couldn't find anything known about the function, we would then give it a name based to say, well, this is part of a family of genes, and these genes are located across the human genome. And this is the page we're looking at here is the HGNC. And HGNC, which is an acronym of an acronym, stands for Hugo Gene Nomenclature Committee, HGNC. And Hugo, of course, stands for Human Genome Organization. And HGNC are responsible for curating the names and functions of the genes in the human genome. And we can see here at the top, I've loaded in gene FAM177A1. There's quite a lot of information associated with it. And first of all, we can see that our gene's located on chromosome 14, Q, so it's on the Q arm. So chromosomes generally have a P and a Q arm. And then this is the actual location itself. But what I just want to draw your attention to is that this gene, FAM177A1, was previously called C14ORF24, which stands for chromosome 14, open reading frame number 24. Again, that's not particularly helpful to understand its function. But what I want to really sort of highlight is that gene names do change over time. And while we don't yet really know specifically what the function of FAM177A1 is, that is likely to change, obviously, as we're all here talking about FAM177A1. And when we find out more about it, HGNC will then probably update this name and give it a name based more upon what we know about its biology, about its function. And you can look at this yourself, and you can link through to all these different databases. You can see we've even got genes in other organisms here. So we've got cow, dog, cat, pig, etc. And this is really helpful because, of course, we can, by looking at these genes in other organisms, we can learn more about their function. So I jump to my next page, and this is a public genome browser, which is maintained and developed on the Genome campus near Cambridge in the UK. And it's called Ensemble. And what you can do is you can load it in. You can see the address up here. And if we search for FAM177A1, we can see that it comes up with lots and lots of information. But first of all, at the top, we can see which is a really fancy, long, complicated looking string of numbers and letters. But really, this is a bit like, if you can imagine a vehicle number plate, which is unique to your car. In many respects, this is the same. It's a unique identifier to identify your gene in this public genome browser. And if we click on the numbers here, what this shows is, first of all, at the top, it's on chromosome 14, our gene, and it's at this position here. And if we look at this, this long image here, this is a graphical representation of chromosome 14. We've got our P arm here, and then we've got a centromere. So this is the sort of the bit that separates our two arms, and this is the Q arm all the way along here. And we see our little box here. This is the location of FAM177A1. So this is sort of an overview of the whole chromosome. And then this little red box, we can then look in a little bit more detail. And this is the genomic neighborhood of genes that are surrounding our gene FAM177A1. I could go into quite a lot of detail here, but I think suffice to say that if we scroll down, we can see the representation of our gene in the human genome. And when I said that whole exome sequencing looks at the bits of the genome that are responsible for making protein. You can see here there are these. This is a representation of FAM177A1 and the various different transcripts that make it up. And the bits in red, these little bars here and here, those are responsible for making the protein. There's quite a bit more process that goes into it, but in essence, you can see that while all of this is the bit that makes FAM177A1, the bit that actually makes the protein are these little bars here. So what I did previously was to load in another gene that's part of the so-called FAM set of genes, and that's FAM176A. And what I wanted to show is that there are these FAM genes that probably go from one all the way up to a number of, sort of a few hundred or whatever. But that this FAM176A gene, as it was, now actually has a new name called EVA1A. So what this shows is how over time a gene that has no obvious clear function can change, and this would have been done by the HGNC group. Now, if we think about Charlotte and Cooper and whereabouts their variants are in their genome, you can see on the FAM177A1 page, we have the coordinates listed here, and I've put those into the Ensemble browser, and I've searched for those specific sequences that are missing. And what we can see here is the representation of our gene at this part of the genome here on this chromosome. And we can see that this is one deletion here. And then this is another part of the genome that's been removed here. So there is a significant part of the genome that's been changed in Charlotte and Cooper. And while mum will have one of these deletions, as I mentioned, or dad will have another, I'm not entirely sure which one is which. But when you have two copies of the chromosome 14 and only one is damaged, that's fine. You can generate enough protein to compensate. But when you have two, you essentially have bits of you're essentially missing two functional copies, and that is responsible for the phenotypes that we see in Charlotte and Cooper. Now, how do we know when a variant is pathogenic? How do we know when it's actually responsible for the phenotype we're looking at? Because we all have lots and lots of different variants in our genome, and some of those will be in genes where they have absolutely no effect on the protein at all. And to come up with an idea of how to actually classify different variants that we find, the ACMG, American College of Medical Genetics and Genomics, have come up with what we call the ACMG guidelines, which allows people to look at a variant and then go through a sort of a rigorous set of classification to see if that variant is likely to be pathogenic or not. And I've loaded up here an article in Nature, which I'll share later. But there's a really handy table in here which shows a number of different things you need to consider, whether that's population data. So, for example, if you look at your patient and you look at a variant and you think, well, that's in the right gene for this this disorder, but then you go and look in the general population, you see, well, everyone's got that, but everyone seems to be fine. So you would think, well, that's probably not the variant. That's probably not going to be causative. Likewise, if you do the same and you see your variant in a gene, but but nobody else has that, then you begin to think, well, that is very suspicious. And there are a number of these different considerations for generating enough support to say that your variant is either benign or pathogenic. And you can see here that the evidence for considering something to be benign is listed here. And if you think whether your variant is pathogenic, you would then have to consider all of these different things through here. And then finally, I wanted to talk through, just briefly talk about Nomad browser. And Nomad gives one of the indications, as I mentioned, for the ACMG guidelines. It allows us to look at a variant in a patient and then compare it to a very large data set of people. So we can begin to build up an idea of how frequent is our variant, and therefore how likely is it that we're going to see it in the general population. So I have done quite a lot of filtering for this, but what I've done is I've loaded in gene FAM177A1, and I've wanted to select all the pathogenic or pathogenic loss of function variants that are contained in this huge data set. I believe it's around 125,000 people. And what we have here is we can see of the loss of function, and of course we believe that FAM177A1 is the phenotype is a result of loss of function, because that's why we're looking at this. There are I think it's 26 I counted loss of function variants. Now, if you count them all up, you'll actually find it's more than 26, or maybe but these ones at the very bottom, these are very low confidence, so we would ignore those. We would ignore those because we don't know if they're sequencing artifacts or what have you. But if we think about how often we're likely to find these variants in the general population, so if we think of 125,000 people divided by 26, that gives us roughly, it's not, I know you'll be on your calculators doing this, but it's roughly 1 in 4,500. So that means on one chromosome, for having one chromosome with that variant, it's 1 in 4,500. And of course, if you have the other chromosome, it's 1 in 4,500 multiplied by 1 in 4,500. And of course, the chances of, if we think about Mendelian inheritance, there's a 1 in 4 chance of inheriting both of those chromosomes together, and that gives us a very, very small number. It's less than 1 in a million. It's actually considerably more, but because of the nature of the database, because the numbers are reasonably small, and we don't quite know how they were generated, we don't know if there was an overcompensation of people with FAM177A1, we would be cautious and say that the chances that the chances of having that combination of alleles is more than, sorry, so it's less than 1 in a million. So while Nomad, it can be very helpful, it's also important to be a little bit cautious about that, but it gives us a rough indication of the prevalence of the disorder. And of course, please don't forget that there are other databases out there. There's Biobank, UK Biobank, there's 100,000 Genomes Project, all of us, and these are really important databases where we can sort of build up more information to work out how often we see FAM177A1 in the general population. Okay, right. Well, I have to go now. That's quite a bit longer than I thought, but anyway, I will leave my contact details with Jill. So if you want to get in touch with me, please do, and I'd be delighted to have any questions about what I've mentioned. So please feel free to contact me. Okay, thanks very much then.